Dr. Geeta, we can start.
Okay. Good afternoon, everyone. Welcome to this webinar series by CSIR, brought to you on Exploring Science with Shanti Swarup Bhatnagar awardees. As you know, we've been bringing these seminars with the popular science talks by the scientists who have received the Shanti Swarup Bhatnagar Award this year. In this series, you have heard a couple of weeks back from the winners in biological sciences and also a winner from medical sciences. So today we have two speakers with us, the two awardees for chemical sciences, Dr. Jyotirmayee and Professor Subi Jacob George. So firstly, hearty congratulations to both of them on my behalf and also from entire CSIR. As you all know, it's one of the most prestigious awards in India. And I think this is an effort actually to see some of the best of the science and take it to the common people as to understand a flavor of the science which is going on in prestigious research laboratories. So this is a small effort on part of CSIR. So today, unfortunately, we do not have DGCSI Dr. Shekhar Mandi, who was supposed to join us today and give his opening remarks. He had some urgent work, but he would like to convey his congratulations and best wishes to both the awardees. And we have with us Dr. Chandrasekhar Srivari, who is the director IICT, who will take us through this event and introduce the speakers. Just a brief introduction to Dr. Chandrasekhar Srivari. He is a very well-known organic chemist in the country and also globally. And in fact, some of you may have seen that there is recently a list with 2% of global top scientists, and Dr. Chandrasekhar has featured in that in organic chemistry. And he is, after obtaining his PhD and postdoctoral work in US, he joined as a scientist and risen to the ranks of director in IICT. He's a member of all the 3 academies of sciences, and he has won many prestigious awards like the Infosys Award and Technology Awards, to name a few. He has several hundreds of publications and lot of technologies he has developed. And some of you may know that in the recent COVID fight, also IICT and Dr. Chandrasekhar have played a very critical role, and they have brought this drug Favipiravir into the market with the industry partner Cipla. So I think chemistry is a very important topic to all of us, and I request Dr. Chandrasekhar to introduce both our speakers and also give his opening remarks and conduct the session. Thank you, Dr. Chandrasekhar, for being with us, and over to you.
Thank you, Geeta, and welcome to our friends Subi and Jyoti. Congratulations again on behalf of the entire CSR family and DGCSIR. Also, greetings to your family on this important event. It's a very milestone day for all of you that both of you have been chosen for very prestigious award of the country. And of course, awards don't come with just cash prize and recognition, but also come with a lot of responsibility. And I'm sure this award will feel more responsible towards science and applications of science towards human well-being. You all have been witnessing the importance of science today while we are fighting COVID, as Geeta mentioned. And also we have been following the statements of Honorable Prime Minister on Atmanirbharta. And as Geeta mentioned, chemistry becomes the central science. And everywhere you see chemicals, including your DNA, RNA, and your inorganic materials and metals and whatnot. So I think we are all proud that we belong to chemistry community, and we all know the tagline of American Chemical Society, proud to be a chemist. So I think we're all proud to be chemists. And again, to take the commitment of Honorable Prime Minister, if chemical sector becomes Atmanirbhar, I'm sure the whole country becomes Atmanirbhar. All your agrochemicals, pharmaceuticals, or diagnostics, I think everywhere chemicals are there, and I'm sure leaders like you would make sure that in the next 10 to 15 years, India would take the position of the best countries in chemistry, beating our neighbors, and we truly become Atmanirbhar. Congratulations again on behalf of all of us. But before we jump on to your presentations and also to interact with the colleagues who are on YouTube and also on the live stream with the CSR portals, I would just quickly introduce the award and also on whose name we instituted this award, and also recollect the great contributions of Dr. Bhatnagar. So I would quickly share 2 or 3 slides, and then from there we will open the forum to both of you. So just to quickly, uh, can you see my slides?
Yeah, we can see.
Yeah. So if you look at Dr. Shanti Swarup Bhatnagar, again, all of us look at Google Doodle, and if someone's name appears on Doodle, we feel that it's a big honor. But the scientists of my generation, if you want to call, I think if a stamp is released on someone's name, that's the biggest honor one could think of. And you could see there is a stamp on the name of Dr. Shanti Swarup Bhatnagar. And if you look at the description of him on this slide, which I took up from Wiki and other information, he is revered as a father of research laboratories in India, and the CSR is because of him. And I know both of you are the beneficiaries of CSR fellowship. Many of your students have got fellowship, and Subi himself was a PhD from one of our CSIR labs in Trivandrum. And again, he has always strived to make sure that how sciences reaches the masses. So his research interests were emulsions, colloids, and industrial chemistry. And looking at his contributions way back in 1928, he has developed a Bhatnagar-Mathur magnetic interference balance in 1928. I think this indicates the passion he has to make equipment for ourselves. While we all talk of Raman spectrometer, I think very few people heard of this Bhatnagar-Mathur magnetic interference balance. And this was marketed by Adam Hilger and Co. in London. Similarly, he always chose industrial problems, gave many solutions to industrial problems for Delhi Cloth and General Mills, JK Mills Limited, Kanpur, Ganesh Floor Mills, Lailapur, Tata Oil Mills, Mumbai, and many things. And his contributions in petroleum industry are also phenomenal. He was the founder fellow of the Indian National Science Academy, and today we all cherish his name. And once his name is attached to any scientist in the name of Bhatnagar Award, I think the glory of the scientist goes manyfold. I think you are very fortunate to be part of this group, and I'm sure you will take his name and fame forward and make India very proud. You all know CSR gives this award every year, Shanti Swarup Bhatnagar Prize for Science and Technology. The purpose you all know, recognition of outstanding Indian work in science and technology. And of course, if you look at Bhatnagar Award, it gives you a cash prize in fundamental science and applied science, in biology, chemistry, environmental science, engineering, mathematics, medicine, and physics. Of course, this all considered for the last 5 years of work, what you have done, but certainly I think the responsibility and the honor you receive by getting this award, I'm sure the next 20, 25 years of your illustrious career, I'm sure you will make all of us very proud. So quickly look at the profile of Subi. Again, a brilliant scientist. I have very personal interactions with him many a times. Very humble scientist. When I recently met him during Siena Rao Saab's 85th birthday, I think, give me a second. I think something went wrong on the computer. Give me a second. Give me a second. Give me a second. Sorry. Can you see now, Geeta?
Yeah, we can see it. Yeah.
I think suddenly there was internet disconnect, and I think there was.
Yeah. Now okay. It's visible.
Subi has been a wonderful young colleague. I met him very recently during Professor Rao's 85th birthday in JNC. Very humble scientist, and I think again and again there's a problem. Give me a second again. Sorry. Yeah, Subi, I think you are doing hide and seek game with me. Yeah, okay. Now you could see Subi on the slide. He got, as you all know, that Bhatnagar Prize for 2020. He was also awarded with the Swarup Fellowship, Fellow of the Indian Academy of Sciences. Then he's on the editorial board of Chemical Sciences, very prestigious flagship journal of the Royal Society of Chemistry. He's on the advisory board of many prestigious journals of IACS and Royal Society of Chemistry, with H-index of 52, 8,000 citations, 130 odd publications. And his contributions are in the areas of bio-inspired fuel-driven living and transient supramolecular polymerization, ATP-fueled enzymatically controlled approaches, then of course chemical reaction-driven strategies, then of course multi-component supramolecular polymerizations, and organic optoelectronics. Again, very proudly, he was a PhD student of our director Ajay Ghosh at Trivandrum Lab. Congratulations, Subi, and looking forward to your talk today. Then we have Dr. Jyotirmayee Das. She's on the call today. Again, congratulations, Jyoti. Again, you have been part of National Organic Symposium Trust as a council member. We work together in conducting many events. She has been fellow of the Marie Curie of University of Cambridge, Alexander Humboldt Fellow at Berlin. Of course, this year, Swarup Prize. She also got Swarup Fellowship in the year 2015. She has over 100 papers. 15 students got PhD with her. Her contributions are in the areas of organic synthesis, developing new methodologies for the synthesis of diverse molecules that we've utilized as lead molecules in understanding the structure and function of therapeutic targets in cellular systems. And of course, she works on non-canonical nucleic acids and supramolecular chemistry of nucleic acids. I'm sure your contributions and also Subi working on the interface of applied science and fundamental science, I'm sure your contributions will take us in a long way. With these little words to both of you, now may I request Jyoti to make a quick presentation to our audience. Just to tell you, Jyoti, our program is currently live webcast on YouTube and also on the CSR homepage. So many young colleagues and CSR family members and many other research scholars are watching. So I request you to start your presentation. I stop my slides here and I look forward to your presentation followed by Subi. Jyoti, you need to unmute yourself. Jyoti, you're mute.
Yeah, good afternoon, everyone. I am very thankful to Dr. Chandrasekhar for his kind introduction. And I am really thankful to CSIR for starting this webinar series, for the idea of this exciting webinar series. And I am honored and glad for this opportunity to present our work on this platform. And today I'm going to give a brief outline of my talk on the topic like application of organic tools for nucleic acid targeted therapeutics. And first, I would like to give an outline of DNA secondary structures. And then I would like to talk about how the DNA secondary structures can synthesize their own selective ligands. So let us first discuss about DNA secondary structures. We all know that DNA is mostly double helical, that is both the strands are held together by a Watson-Crick type of canonical hydrogen bonding, but DNA can also adopt higher order secondary structures. In particular, guanine-rich DNA sequences can adopt 4 standard secondary structures called chick quadruplexes. As a basic feature, they contain coplanar arrangement of 4 guanines that are linked together by Hookstein type hydrogen bonding. The story of chick quadruplexes was started with a bang. It was Dr. Bang, a medical doctor by profession, who became curious about nucleic acids. In 1910, he discovered that 5' guanosine monophosphate can self-assemble in water to give a gelatinous material. Decades later, it was found out that the gel contains like G-quartet-like building blocks, and the G-rich DNA sequences can adopt G-quadruplex structures, and they also contain these G-quartet-like building blocks. Subsequently, it was found out that G-rich sequences are present in the telomeres and in the promoter regions of several oncogenes, and they play key biological functions like telomere length maintenance and regulation of gene expression. The complementary cytosine-rich DNA sequences can also adopt folded conformations called I motifs. The I motifs contain intercalated cytosine-cytosine base pairing due to hemiprotonation of one of the cytosines under acidic conditions. Both G-rich and C-rich sequences are present in the regulatory regions of oncogenes which are responsible for uncontrolled growth of cancer cells. It has been shown that small molecules can bind and stabilize DNA quadruplexes and modulate their expression in cancer cells. Therefore, small molecules that can bind and stabilize DNA quadruplexes are considered as potent anti-cancer agents. In recent years, the I motifs have also emerged as potential therapeutic targets. In our lab, we use classical methods like synthesize large number of compounds and then screen them to identify selective compounds for targeting DNA quadruplexes. And these are nucleoside derivatives, peptide mimics, and small heterocyclic compounds. However, the rational design and synthesis of ligands is quite difficult and challenging because these DNA quadruplexes and I motifs are highly dynamic in nature. And moreover, many reported ligands show poor selectivity for quadruplexes over duplex DNA, which is highly abundant in the human genome. In contrast to G quadruplexes, only a few ligands are known for targeting I motifs. Targeting I motifs is challenging because due to their pH-dependent structural topologies. Moreover, we also wanted to target RNA secondary structures. In addition, many reported ligands are not suitable for advanced clinical trials because many ligands show poor solubility in water, they lack membrane permeability, and many of these compounds have been synthesized by multisteps. So it is important to develop ligands that can be synthesized easily in the lab. And secondly, the selectivity, which is one of the major issues, that it is not only important to develop ligands that are selective for quadruplexes over duplex DNA, but the ligand should show selectivity between different quadruplex structures. So this is required to reduce the cytotoxicity of the ligands in the cellular system and also to reduce the undesired side effects of the ligands in living cells. Now, the most important question is the cellular localization. The ligand should also be trapped, should be easily trapped in the cellular system. And finally, the important question is whether a DNA secondary structure binding ligands, that is, whether a G quadruplex or I motif ligand really alter the transcription of a particular oncogene. We wanted to gain insights into the mechanism of regulation of gene expression by ligand-mediated stabilization of a quadruplex structure. So I talk about the DNA secondary structures, and now I would like to talk about how the DNA secondary structures can synthesize their own ligands by using target-directed synthesis. So what is target-directed synthesis? In general, there are two approaches. One is in situ cycloaddition. In this approach, reactive fragments capable of binding the biological target can come into close proximity and then undergo an irreversible reaction to provide products that can strongly bind to the biological target. The second approach is dynamic combinatorial chemistry. This approach uses reversible bond formation building blocks assembled through reversible bond formation to develop a dynamic combinatorial library of interconvertible products. In the presence of a target, the equilibrium of the system shifts towards those products that can strongly bind to the biological target. So both these approaches can combine synthesis and screening of ligands. Therefore, they provide unique platforms for the in situ synthesis and screening of selective ligands for a biological target. So now let us to begin with, we have started the in situ azide and alkyne cycloaddition to develop selective ligands for DNA quadruplexes and I motifs. So azide alkyne cycloaddition, this is a name reaction which is commonly known as Quiescent cycloaddition. Even it is known as click chemistry. The cycloaddition between an alkyne and azide can provide both 1,4 and 1,5 triazyl regioisomers, either the 1,4 or 1,5 triazyl regioisomers. In target-guided synthesis, which is commonly known as in situ click chemistry, the target's binding site is used to select two small organic reactants and then force them to react with each other in a regioselective manner. So to start with, we have started quadruplex. We have synthesized a pool of alkyne and azide fragments and then carried out the reaction using conventional solution phase synthesis. However, the lead identification was somehow difficult, that we could not identify the lead compound. We could not separate the lead compound from the unreacted starting materials. So in order to isolate and identify the lead compounds in an efficient manner, we have immobilized thiolated DNA templates on gold-coated magnetic nanoparticles. So we have taken target templates like DNA quadruplexes and I motifs, and we have also taken the control templates like duplex DNA and a single-stranded DNA. And these DNA nanotemplates were characterized by using circular dichroism, UV visible spectroscopy, and K imaging. So then this is our method, that in our approach, we have taken a mixture of alkyne and azide fragments and then incubated with the DNA templates. Then after giving sufficient reaction time, we have used an external magnet to isolate the DNA nanotemplate along with the newly generated triazyl products. The unreacted starting materials remained in the supernatant. The ligand-bound DNA nanotemplate was again resuspended in buffer and heated to release the triazyl products, which were characterized by HPLC and mass. So this is the chemical library that we have developed in our lab. So we have just prepared 4 different alkynes and 17 different azides. So the multi-component chemical library could provide 136 possible triazyl products, including syn and anti, that is 1,4 and 1,5 triazyl regioisomers. But in the presence of a target, that is, we could identify 4 selective leads for each of the DNA quadruplexes and I motifs. We have taken 2 different quadruplexes which are present in the promoter regions of CMYC and BCL2 oncogenes and their corresponding I motifs. The CMYC and BCL2 oncogenes play key role in cancer progression. Regulation of these genes by targeting their promoter quadruplex and I motifs could be a potential anti-cancer strategy. So then we have studied the interaction of these ligands with these DNA targets by using a series of biophysical studies. So I'm not going into details of these studies. We have used Foster resonance energy transfer based melting analysis to determine ligand-induced stabilization of a folded quadruplex, isothermal calorimetry, fluorescence spectroscopy, UV visible spectroscopy methods were used to determine the binding affinities of these ligands for the DNA structures, and the mode of binding was studied by using circular dichroism and enamel spectroscopy. So this is the results. The results are consistent with the TDS method, that is the target directed synthetic method, that a lead compound binds selectively to the DNA target that promotes its formation. So then we have evaluated these ligands in the biological biological system by using a series of assays. The cytotoxicity was studied. We have taken both cancer cell lines and normal cell line, and these ligands are selectively toxic to the cancer cells and non-toxic to the normal cells. Only one of the ligand, which was obtained as a lead compound for BCL2I motif, was found to be non-toxic for both the cancer and normal cells. We have also used fluorescence imaging to study the localization of the ligands in the cellular system. So we found that the ligand is cell membrane permeable and can mostly localizes in the nuclei of live cells, and further immunocytochemistry using antibody reveals that the ligand can induce and stabilize the formation of quadruplex structures in the cellular system. And finally, we have studied the effect of these ligands on regulation of gene expressions. Mostly these quadruplexes and I motifs are also considered as genetic switches. So the results, like we have used these assays like qRT-PCR, Western blot, promoter luciferase assays, and which revealed that these ligands can modulate gene expression in cancer cells. So therefore, these ligands can therapeutically modulate cellular processes, and this method can be useful in cases where we cannot target the proteins for undruggable protein targets. So thus the method, the in situ cycloaddition using DNA nanotemplates, reduces the time required for the conventional chemical synthesis and their biophysical screening of ligands. It enables efficient isolation and identification of lead compounds. It also prevents the wastage of expensive commercial DNA targets, as the DNA nanotemplates could be easily recovered and reused for subsequent reactions. And moreover, the resulting compounds can selectively bind to the DNA targets and show potent anti-cancer activities. So now we have used this method to develop selective ligands for the TAR RNA as the template. TAR RNA, that is the trans-activation response RNA, is found in the HIV-1, that is human immunodeficiency virus type 1, HIV-1 genome. And this TAR RNA can adopt a stable hairpin structure, and it interacts with the TAR peptide and promotes viral replication. Therefore, TAR RNA has been targeted by small molecules to inhibit TAR-TAR interactions and thus viral replication. So for TAR RNA, we have developed a simple chemical library consisting of thiazole polyamide-based alkynes and simple azides. And we have used TAR RNA as the target template, and we have used TAR RNA without this bulge region, and a TAR DNA as the control templates. But the in situ cycloaddition, in this case, we have used biotin TAR RNA, and we used streptofidin beads to isolate the TAR RNA. And the method is similar like the previous cases. So by using this method, we have identified a thiazole-based peptide mimetic that binds in the bulge region of the TAR RNA. And further, the in vitro biophysical studies reveal that this ligand can also inhibit TAR-TAR interactions. So the compounds that can bind to the TAR RNA and inhibit TAR-TAR interactions are considered as potent antiviral agents. So I have shown a rapid method to develop TAR RNA selective ligands. The ligands that can bind to TAR RNA and inhibit TAR-TAR interactions are potent antiviral agents. So I have shown that from a pool of alkyne and azide building blocks, the DNA and RNA secondary structures like DNA quadruplexes, I motifs, and HIV RNA can synthesize their own selective ligands. Now, I would like to present the second method, that dynamic imine exchange for targeting a DNA quadruplex. So, in dynamic combinatorial selection of G quadruplex ligands, we have used aldehyde and amine building blocks that reversibly react to generate interconvertible imines. The G quadruplex nanotemplate selects its best imine binders, and then the imine was reduced to give the corresponding amines. Then the ligand-bound amine template was separated by magnetic separation, and then it was heated to release the amine ligands. We have also carried out a control experiment using duplex DNA as the nano template, and we have used a carbazole aldehyde and 10 different amines that reversibly react to generate imine-based dynamic combinatorial library. In the presence of quadruplex nano template, the system evolves to generate quadruplex selective imine binders, and then it was reduced to give the stable amine ligand. So by using this method, we have identified a carbazole amine that can selectively bind quadruplex over duplex DNA and also reduce the c-MYC expression in cancer cells. So this is the first example of an imine exchange reaction using a DNA nano template to develop selective ligands for DNA secondary structure. And so I have shown you both these approaches that we can use in situ cycloaddition to develop selective ligands, and we can also use dynamic combinatorial chemistry to develop selective ligands. In the first case, we have used alkyne and azide fragments, and the DNA nano template selects its own alkyne and azide fragments to give the lead compounds, and in dynamic combinatorial chemistry, the simple building blocks assemble to give a reversible dynamic combinatorial library. That is, all possible products are first formed, and the target then selects its best imine ligand, which were then reduced by sodium cyanoborohydride to give the corresponding amines. So in this case, we could identify triazole-based ligand, and in this case, we have identified the amine-based ligands. And both the cases, the method combines synthesis and screening, and the DNA templates could be easily recovered and reused. So finally, I would like to give you that G-quadruplex DNA interactions are not only useful for therapeutic applications, they can also be used for constructing biomolecular devices like transmembrane ion channels. So ion channels are pore-forming proteins. They are present in the lipid bilayer, and they allow passage of ions across the lipid bilayer. So they play an important role in nerve impulse, muscle movement, and other biological processes. Since the G-quadruplex DNA is stabilized in the presence of metal ions, we wanted to explore this property to develop ion channels to mimic the transmembrane ion channels. However, the insertion of negatively charged DNA into hydrophobic lipid membrane is energetically unfavorable, and moreover, the design of a selective DNA-based ion channel is challenging. So, in the literature, DNA origami, that is folded DNA structures, and double helical DNA-based ion channels have been reported, and in these examples, the DNA is linked to porphyrin through lipophilic linkers. Porphyrin stabilizes the DNA, and the lipophilic linker promotes the insertion of the DNA into lipid membrane. In our design principle, we have synthesized a liponucleoside derivative, that is a lipoguanosine derivative. The guanosine derivative can stack upon the G quartet of G quadruplexes, and due to the presence of lipophilic side chains, this ligand can also promote the insertion of the DNA quadruplex in the lipid bilayer. And finally, using biophysical studies and theoretical studies, we have shown that these three molecules of this ligand can bind to the DNA quadruplex, and the DNA quadruplex and the molecule assembly can form channels in the membrane, and they can selectively transport K+ ion across the biological membranes. So we have also developed DNA-based logic devices by using enzymes. So in these cases, we have taken a small molecule DNA quadruplex and nucleus enzymes, and the method was also useful to program a device for determining non-zero square numbers up to 20. So finally, the future perspectives include rapid synthesis and high throughput screening of ligands for new and challenging targets, for targets whose structural details are yet unknown, and also it can be used for targets where rational ligand design is difficult. And moreover, it is important to develop new bioorthogonal reactions for generating more specific and more complex ligands. And it is also important to explore small molecule nucleic acid interactions for creating smart biomaterials with applications in nanotechnology. So in our lab, we integrate chemistry, particular organic chemistry principles with molecular biology and nanotechnology. Chemistry, as has been told by Dr. Chandrasekhar, is the quintessential molecular science that can be used to create new molecules, new materials, new tools, and new theories that can find diverse applications in materials, nanotechnology, environmental science, and life science. In addition to targeting nucleic acid in our group, we also work on DNA-based biomolecular devices, and we also use nucleic acid components like DNA and RNA, nucleosides, and nucleic acid derivatives to study their self-assembly process for drug delivery applications. And as trained as an organic chemist, we also develop new methods to synthesize biologically active natural and non-natural compounds. And finally, I would like to thank my research scholars for their hard work and cooperation. And the research program has contributed to several drug-like candidates, interesting biomimetic systems, and also new organic transformations. And I would also like to thank the funding agencies like DBT, Wellcome Trust, DST Swarna Jayanti, CSR, DAE, and DBT JNCASR for funding. And I would like to thank you all for your kind attention. Thank you.
Thank you, Jyoti, for a very wonderful lecture, and I'm sure it will attract a lot of questions from many young colleagues who are live watching on this. So we will hold the questions till end of Subi's talk, and then we will take the question answer session. Again, wonderful talk, Jyoti. Congratulations. May I request Subi to go ahead?
Yep. Can you see my slides?
Yes, Subi, we can see you.
Yeah. Good afternoon, everyone. At the outset, let me thank Dr. Chandrasekhar for a very kind introduction. And also, I take this opportunity to thank Dr. Mande and entire CSR family for giving me this opportunity. As Dr. Chandrasekhar mentioned, I'm very grateful to CSR from my student days, starting from my fellowship to do PhD, and also for a kind of very successful PhD in a kind of CSR lab under the supervision of Dr. Ajay Ghosh. So always there was a connection with CSR, and so very happy and very humbled and proud to be here. So today I'll be sharing some of the kind of research field we are doing, probably considering the diverse audience. I will not be going into the details of my research, but on the other hand, the way we are thinking how we would like to push our research field is probably I will be sharing with you. And let me tell you, I am a kind of supramolecular chemist and a material chemist. So that means we work beyond the molecules, like we start from molecules and try to assemble them to make large assemblies with the ultimate aim of making interesting materials. And most of the time, our inspiration comes from biology, okay, our biological world. And that is something which is true with not only me, with most of the supramolecular chemists. You can see that there is a nice kind of animation just to give you a picture of the kind of materials we are working on. Basically, these are kind of supramolecular polymers, and I'm sure all of you know what is a polymer. They are large macromolecules which has kind of repeating units or monomeric units, and you know that without polymers, we cannot imagine a world right now. Our life is impossible without polymers right now. So you can see that only difference here. I will come back to the little bit detail later, but just wanted to understand the kind of what is the power of a molecular assembly, or why we want to work with assemblies. You can see that this is, if you look into the structural wise, it's very similar to the classical polymers in terms of its long chain structure. But only difference here is you can see that the monomers are linked together by very weak interactions. So weak interactions like many things in nature, like hydrogen bonding, pi-pi stacking, or so on. So as a result, what happens is, although they structurally very similar to the classical polymers, the monomers are dynamic in the sense monomers can come out and go in. So in other means, I mean, we can say that there is a monomer exchange dynamics. So as a result, we can think that this kind of materials can be easily reversible because these are weak interactions. We can break them. They are adaptive. They are dynamic. So probably we can think about many properties which are not able to provide by the classical polymers. Okay, this is, let it be like a kind of preface. Let's move on to the real inspiration. I'm like, as we talk, most of the supramolecular polymers, they say that their inspiration comes from biology or biological world or cellular world. We know that we cannot make a life or even the smallest component of a life, that is a cell. If you look into it in terms of complexity, probably this is the most complex structure, even compared to a galaxy, or irrespective of its size, is probably one of the most complex structure. So probably the, let's not mistake me, I mean, in the sense, the objective of supramolecular chemistry is not to create an artificial cell, but the objective is to probably learn from cell, look into its multiple components inside the cell, and probably try to mimic in a kind of synthetic material. And interestingly, we can think about that. We can make interesting material with dynamic, very interesting properties. Okay, I'll come to that very soon. So basically, what we are trying to do, we start from molecules, we try to assemble them. And you know that organization is an important parameter both in cellular world for its function. Okay, so what are we trying to mimic? So if you look into the cell again, there are two interesting structural aspects to it, right? If you look into it, it's most of the functions, like if you look into the motion, muscle action, anything, if you look into it, the structure is not most of the time is driven by single molecule. So let's look into the cell membrane, protein assemblies, or DNA. So there are more than one molecule, right? So that's probably kind of first structural lesson we can learn from nature. So it's always more than a molecule. It's a kind of assembly of molecules doing the job for this particular cellular world. And also another aspect which we are something which it's intriguing as is kind of is dynamic world. So if you look, this is a kind of YouTube video. You can say that this is a microtubule protein, which is very important for our muscle movement and so on. And if you look into it, this is again a kind of assembly. It's not a single protein. Large number of protein monomers are assembled together, and they form large structures. Okay. And they are continuously assembling and disassembling. This is a nice video. You can find it in YouTube. It's called Inner Life of a Cell. And you can see that they are continuously assembling and disassembling. So the dynamics, so like a kind of, we can say that cells are always, we are always not at equilibrium. We are always moving. We are always out of equilibrium. So if you look into these two aspects, one is that organization and the other one is a kind of dynamics. Can we mimic in kind of synthetic materials? And probably that's the first motivation for a supramolecular chemist to come with a field called supramolecular chemistry. And if you look into the organization aspects, the first aspect of it's a kind of more than one molecule, how to assemble molecules. So chemists basically learned that we can use very weak non-covalent interactions. Again, like in nature, like DNA, you know that it is stabilized by hydrogen bonding and pi stacking interactions. So similarly, we can use these weak interactions to assemble molecules. That's probably like 3 decades back, the supramolecular chemistry field has come up and it got Nobel Prize in 1987. And as these Nobel laureates say, they defined this field as kind of chemistry beyond the molecule because this is not individual molecule. It's actually an assembly of molecule which is doing the job. And if you look into it, often they say that in nature, you have wonderful examples of substrate protein complexes. So there is a molecular recognition happens. So can we make synthetic mimics of that? And that is what probably the first host case complexes like receptors, they all came up like a crown ether, we know that, which can bind to metal ions. So which were all designed based on molecular recognition, right? What about dynamics? So this is only a kind of assembly of molecules. Can we go ahead and think about dynamics? And again, I'm sure all of you are aware of like a very 2-3 years back, the 2016, the Nobel Prize has gone to molecular machines for these 3 gentlemen, Sauvage, Stoddard, and Feringa. And probably this is a first attempt chemists have made to mimic motion, right? Or mimic a movement. So this is molecular machines. And again, the motivation, you know that in biological or cellular world, there are large number of examples like a protein assemblies and also the bacterioflagellum. They're all continuous molecular motor and they're continuously moving. So can we make something synthetically with such molecules? And that is again a kind of branch of, it started as a branch of supramolecular chemistry. And you can see that these are some of the examples. I don't want to go into the details where they clearly show that molecule can indeed perform work. And always they need a kind of fuel or energy for the molecules to move. And you see that this is video is from a Ben Feringas group. This is a kind of wonderful demonstration. What he has done is kind of arranged a kind of large number of molecule on a surface, which is a molecular motor triggered by light. If you shine light on it, it will start rotating. So you put a kind of a small piece of glass nanorod on top of it, and it starts rotating. Okay, when you shine light. So that is probably the first macroscopic observation that molecules can indeed perform work. And although the real use of this molecular machine in terms of performing work is still probably many years away, but this is probably the first attempt from the chemist side to really mimic the molecular motions. Okay, but then let's move on. This is not what we are doing. Sorry. Yeah. So if you look into, again, coming back to this YouTube video, which is a kind of inner life of a cell. If you look into that, you can see that it is not again at a molecular level. It is very high, kind of higher hierarchical level. You see that it's a kind of probably micrometer long protein assemblies, which are continuously assembling and disassembling. So the assembly and dynamics probably is happening at a higher length scale. So probably that is the inspiration. If can we mimic such kind of protein assemblies which are continuously assembling and disassembling, and if you are able to do that, probably that's a way to make a kind of dynamic material, right? And that is probably the kind of topic we were working on, supramolecular polymers. And you can see that this is again large molecular weight, and it has molecules which are linked by kind of non-covalent interactions. The monomers are a kind of continuous exchange. So they are kind of assembling, disassembling. And probably this is a kind of structural analog. I'm not talking about in terms of function, but in terms of a structural analog, probably this is a wonderful example of protein assembly, right? But obviously the functions are completely different we are going to talk about. So this field is quite active for probably last 2 decades. And I had the opportunity to grow along with the field. So what we have been trying to, probably over the 2 decades, people have found that this is very similar to the classical polymers in terms of mechanisms and so on. But then it has interesting properties like dynamics properties and so on. So what we have been trying to do is to control its structure, to control its sequence, control its kind of what happens if we go from one monomer to the multiple monomers, and can we also go to out of equilibrium? That is something which is nature has or cellular world has. So these are the few attempts we have been trying to do it over the last probably 10 years or so. Let me just tell you this slide because this is very important. In 2020, it's a very special year for polymers. All of you know that this is 100 years of polymer chemistry. In 1920, this gentleman, Hermann Schrödinger, really proposed what is kind of a polymer. This is a classical paper. He published that polymer is indeed connected by monomer units, and they are large structures and really linked by non-covalent bonds. And So just wanted to probably remind all of you this particular occasion because it's very appropriate. Probably 100 years after 100 years, we are talking about this new kind of material which is called as supramolecular polymers. So let's come back to the supramolecular polymers. I'm sure all of you are familiar with this particular movie, it's a Terminator, and you know that he has a wonderful talent to self-heal, right? And even if you try to destroy it, it has a nice tendency to come back and self-heal system. So that is probably one of the properties we can expect from the supramolecular polymer. So even if you cut it into 2 pieces and put it back, and because of the dynamics of the molecules, and they can self-heal and go back to the original material. And that material will show exactly similar mechanical properties before cutting. Okay, so isn't it nice that the dynamics of molecules in a material is bringing back the interesting self-healing properties? Okay, so this is one of the probably the most look after kind of property of a kind of polymer. Okay, and this self-repairing. You know that it is quite common in biological world or cellular world. And by making a supramolecular polymer, probably we can kind of extend into a kind of synthetic material where it has kind of self-healing properties. So another one thing, I'm like, this is something general about supramolecular polymer, and more specific about what we are trying to do is, if you look into biological structure, there is one more interesting aspects into it. That is because always we hear that be at equilibrium, that is very important. But in terms of biological structures or cellular world, we are never at equilibrium, right? So it's a simple energy landscape, you can see it. And most of the synthetic system or synthetic material, when you make it, and especially when they are linked by very weak interactions, what happens is, since they can easily form and reform again, always you end up in a kind of thermodynamic equilibrium. Okay, even if you get trapped in a kind of kinetic trap or something, this dynamics will help the system to come back to the thermodynamic equilibrium. So it's a basically a kind of energetically kind of gain in free energy drives the formation of the material. But if you look into the most of this biological world or cellular world, something very interesting is, although we have examples of kind of thermodynamically stable assemblies like cell membranes, but if you look into this protein assemblies, again, the microtubules, they are never at equilibrium. They are coming under this side of the energy landscape. That means it's out of equilibrium structures. You need a kind of continuous supply of energy for the system to remain in a kind of assembled state. And once this fuel or energy runs out, the system relaxes back to the thermodynamic equilibrium. And this is very important. This functional state, which is away from equilibrium, is very important to perform most of the important functions of the cell, starting from cell motility, movement, signal transduction, everything these out of equilibrium structures are doing the job. So from a materialistic point of view, can we make such out of equilibrium materials? This is another aspect we are interested. So supramolecular polymers, can we control its structure? Can we control its temporal properties? What I mean is, a kind of out of equilibrium material like this has a kind of temporal properties. It forms, deforms with time. That means the complexity of that organization changes with time, right? Unlike these thermodynamically equilibrium materials. So these were a few things we wanted to make. We wanted to learn from biology and to probably take that as an inspiration to make synthetic materials. Let me correct again. At any point, we don't want to make a cell. Just cell is an inspiration for us to learn many lessons and trying to make materials. Okay, so let's move on. So how do we do it? So from a chemist point of view, if you want to structurally mimic a kind of temporally or out of equilibrium material or structurally controlled material, let's look back to how nature does it again. As I told again and again, inspiration comes from nature. This left side video is a kind of microtubule self-assembly, and right side video is a kind of actin self-assembly. So both are kind of protein assemblies, and microtubule, and both are very important for many functions in the cell, as all of you know. And it works on a very simple principle. So you can see that there are monomers here, but monomers remains in a kind of inactive state. They will never assemble unless you trigger it by a kind of chemical reaction. Okay, and in this case, if you look into microtubule, it's a kind of GTP binding, and if you look into actin, it's a kind of ATP binding, right? So it remains in a kind of dormant or inactive state. Once one of these binds or a fuel binds to it, there's a conformational change. It changes into a kind of active state, which start growing. And there will be obviously, once ATP or GTP get converted back to the ADP or GDP, it disassembles the structures. It is much more complex than I explained, but from a chemist point of view, let's very simplify this, and let's think about how we can make such a material in synthetically. And you know that you need a fuel, and we can, as a chemist, you know that there are various kinds of fuels are available, like enzyme. We can use a chemical reaction, or we can use a kind of redox properties, and so on. So this is something which we were trying to do, kind of fuel-driven assemblies of biological structures. Can we structurally mimic in kind of synthetic system so that we have a control on its structure, and also can we have a control on its temporal properties? Okay, so that's something which we were trying to do. These are a lot of molecular structures. I don't want to go into it, but just see that we have used various fuels. And one example I want to show, just by schematic, you can see that this is a kind of very simple molecule. It has phosphate receptors at both ends. And this is a phosphate receptors where we plan to, where ATP can bind selectively. Okay, so if you couple this self-assembly of this molecule, so this molecule will remain in a kind of inactive state, just like actin. But once ATP binds to it, it can start assembling and form a supramolecular polymer. And once ATP start hydrolyzed to ADP, it can disassemble. So what we have done is we have kind of this ATP ADP reaction cycle, we coupled to two enzymes, phosphoryl transferase enzymes. So, and we control their concentrations, both kind of primary fuels, enzyme concentrations and everything. So basically we have recreated kind of structural mimic of actin. Not again, let me correct, it's not functional mimic, it's a kind of purely structural mimic. And we could create almost all the kind of structural aspects of in terms of its self-assembly, in terms of its structure, in terms of its temporal aspects, we could create. And so we have recently extended it to chemical reaction driven, redox driven, everything. Don't worry how we did it, because it's a kind of general talk. I don't want to go into it details, but what we achieve from that, why we want to make that such biomimetic materials. One of the striking similarity we found that all of them follows like a kind of nucleation growth assemblies. Most of these protein assemblies has a kind of nucleation growth assemblies, and we found that all the system shows that kind of nucleation growth assemblies in terms of structure, and which is controlled by the fuel concentration. So rate of the growth of your assemblies can be controlled by fuel. And you know that chemists are clever. This fuel concentrations, we know we can couple to external chemical reaction networks, and that way we can control their rate. So what we could achieve as a result is we can make systems which has a controlled length, because fuel determines the length, concentration of the fuel determines the length as well as the rate. So we can keep adding monomers into the system starts growing. So we can have a control on their molecular weight. And then just like natural systems, they are monodisperse in nature. Dispersity is one of the issue with most of the materials we make. We are very structurally narrow, I mean like a kind of monodisperse. That's another advantage we get it. And one of the most important advantage, I feel, kind of new kind of materials we can think about. We call it as a transient material. And you see these are the two phosphoryl transferase enzymes. We couple this reaction network, and you see that you can create a kind of material which is stable only for a kind of definite lifetime, which we can define. Okay, so that means that's why I call it as a transient material. Transient materials are those materials which has a kind of definite lifetime and something which we define, and or something which we can pre-program, right? So what happens is, you can see that this just, I would like to draw your attention to this particular graph here. You can see that this is the lifetime of a material, and by controlling the fuel concentrations or the many variables are there. I am not going to the details, enzyme concentration, anything we can vary. So basically what we are ending up is a kind of materials where we can change the lifetime of a material, or we can predefine the material, and we call it as a kind of transient material. It's exactly what happens in nature. You see that assembly forms, disassembles later. And only difference is, if you want to repeat this cycle, as you can see here, we are not as clever as nature. You have to add another batch of the fuel. Obviously in nature, you see that it is all compartmentalized. This cycle happens continuously. But our case is, unfortunately, it's not a closed system. It's an open system. So we have to probably add at the end of each cycle. This is something aspect probably one has to improve. We are also thinking about it. But the only thing is that from a material perspective, that's not really matters, right? Why we want to make transient materials? It's a kind of, you can see that some of these things are very familiar to you. If you watch Mission Impossible, you know that after the secret message, the kind of the device just disappears, right? It just self-destruct itself. So one can imagine that if you create a kind of transient circuit, we can probably make kind of such devices, or self-erasing inks, or just like nature, you can think about spatial temporal patterns which are completely driven by chemical reaction. You can think about a drug carrier drug delivery carrier, where you, instead of classical stimuli responsive where you need another stimuli to break it, you can think of probably predefine the lifetime of a material. Okay, don't ask me, none of them are really realistic right now, but this is probably, one can imagine this probably a kind of scientific fiction. We can probably extend this out of equilibrium chemistry, what we are doing, which is fuel driven. Probably we can make materials which are kind of pre-programmed lifetime. Okay, it's up to your imagination. You can probably think about very interesting material. But most important thing is, from a material point of view, through this bio-inspired strategy, we are able to make supramolecular polymers which has a definite structural control and also temporal control. And kind of applications we can always emerging, because still we are in a kind of, we could not get complete control on structural aspects. People are working on this various aspects of it. So what is next? I think I have 5 more minutes, so I will try to see that So we are able to make chemical fuel driven strategies. We are able to make structures which are well defined. They are temporal. And what about function? So one of the issues is like if you want to mimic some of this protein assembly's function, one of the important thing is a kind of motion. Can we control this assembly disassembly pattern into a kind of movement? This is something which we are now trying to work on it. For that, we need to kind of store the energy in the system. Unlike the so far the system I'd shown, where if fuel acts, it disassembles immediately. But for this to act, you know that microtubules have a kind of very characteristic nature that disassembly is catastrophic. That means there is a strain buildup in the system and just burst. And that energy release is helping to movement and so on. So we are now trying to create kind of kinetic bottleneck in the disassembly pattern, and we are working on it. And we see that with this complicated molecules, we are in a kind of very recent results. We are kind of, this is the disassembly, and you can see that it's extremely fat or highly cooperative. So we are able to kind of catastrophic disassembly could be mimicked in this kind of material. This is very ongoing projects. And you see that these are, you can see something is moving. These are kind of red fluorescent beads, silica beads put on these fibers. And you can see that this assembly disassembly process coupled with these patterns are indeed able to generate a force, and these things started moving, this kind of beads started moving. We are now trying to quantify the kind of how much force is involved. But the message I wanted to say that you can see that biocellular world is a kind of good kind of learning lesson for us. How to mimic these biological structures, we can create interesting materials. And so this is something which I already told. And the final thing is, what we are now trying to do is, can we also do the sequence-specific polymerization? You know that nature does protein synthesis in a kind of highly sequence-specific polymerization using various amino acids and so on. Can we do it using the supramolecular polymers? I know that it is a big challenge again, because as soon as you think about a two monomeric system, there are large number of possibilities you can think about it. You can think about they can completely phase separate. They can make interesting axial heterostructures like this is like inorganic heterostructures. They can have this kind of AB kind of organization and so on. So if you can make some of these structures like axial heterostructures, probably that is very similar to a competition, good competition to the inorganic material chemists, because these kind of axial heterostructures are very important for kind of photocatalysis, energy storage, and so on. So we are thinking, this provides us a tool to make kind of organic heterostructures if we can control this sequence. And something I would like to mention is that most of the lessons we learn although we get inspiration from biology, which has a very good similarity with classical polymers. Okay, so we can look back to the classical polymerization, how people have done copolymerization and so on. For example, you know about the reactivity ratio, how strong AB interactions happens. That is very important in determining the sequence of these classical copolymers. And that's something we found that almost similar principles can be applied to the supramolecular polymers as well. And we are able to, you can see that this beautiful super resolution microscopic images, you can see that we are able to make this kind of axial organic heterostructures. And these two monomers are kind of optoelectronically active complementary systems. And this gives us this kind of structures we could not achieve before because of the lack of understanding on its mechanisms. And now we are in a position to go from one component to two component and to make these axial heterostructures. And it gives probably interesting opportunities for us to look into the exciton migrations from a materials perspective of point of view. I do not want to go to the details again. So what during the last probably 25 minutes, what I was trying to show you is we are trying to learn the principles of molecular self-assembly by learning lessons from nature. We are trying to go more and more complex systems, multi-component systems, trying to move away from equilibrium to out of equilibrium to make transient materials. And obviously, once we learn enough, we should convert into a kind of interesting functions. So this is basically we have been trying to do probably last a few years, and I hope I could convey in a kind of simple language. We have a lot of challenges ahead, and as soon as we realize that we go to kind of monomer sequence and multi-component systems, obviously the characterization tools have to be improved. We need a kind of predictive synthetic design if this field has to move forward. It's very important because whatever we are now trying to see is probably one system it works very well, probably in the second system it's not so accurate. That's an important aspect in the supramolecular polymers field. We should have a predictive synthetic design. And something which is important, we think, because we talked about polymers a lot, and at the same time, we know that plastic waste is an important concern. And at least there is a community which seriously think, since supramolecular polymers are completely recyclable, we can take back the monomers by breaking the weak bonds. Probably it's an ideal material for a circular economy. Probably go back to the monomers, reprocess it again. So we don't have to even think about probably biodegradable polymers, because biodegradable polymers also probably take long time. Probably that is one way people are thinking. Anyway, we are not working towards that direction, but that is probably one opportunity for the supramolecular polymers to go back to reuse the entire monomeric systems and so on. We have to. This is something which aspect I don't want to elaborate, but then we are now trying to think about secondary nucleations and so on, which is again a well-known principle in biological systems that gives us a kind of a tool to control the topology of the systems and so on. With that, I would like to thank, these are my PhD students, along with a lot of postdocs, R&Ds. I would like to really thank them because whatever I achieved so far speaks the volumes of their hard work and dedication. Thanks to all of them. Thanks to all my collaborators, and also funding comes from various sources. And more importantly, thank you, all of you, for listening to me. Thank you.
Yeah, thank you, Subi, for a wonderful presentation. I think maybe you could stop sharing the slide. Thank you.
I did it. Yeah.
Super, yeah. Okay. Thanks again to both the speakers for joining our CSIR invitation to take science with the masses, if you won't really call it. And I'm sure this YouTube will be there, and many colleagues will keep watching it, if not today, tomorrow and all. And I could run through the YouTube link and on the Facebook, and a few questions are getting typed. So what I would do is maybe I'll take first questions for Jyoti, because she was the first to give the talk. And then maybe Subi, I'll take questions to you. But there are some common questions which I think have come from some school children or some young colleagues, actually. I mean, you're already young, both of you, but younger than you guys. So maybe someone who has just begun their career. So Jyoti, you have one question from a colleague. Can your work be useful to COVID diagnostics or treatment?
Yeah, since we target nucleic acids, so if we can treat the nucleic acids of the COVID, SARS-CoV-2 virus, the RNA, then we can selectively target the RNA of the SARS-CoV-2, which is creating the disease. So in that way, we can modulate the function of the SARS-2 COVID-2 virus, and we can develop drugs for corona. But this is a long-term process. Like, we have to use those RNA, which we have started already working in that, and we have to use that, like in our method, that we use that viral RNA and do the synthesis of ligands. Since it's the modulation of nucleic acid, one can do that using our chemistry.
The question is, are you working towards this, because this has become a pandemic and you would like to have some...
Yes. We started working on this.
Okay. There is a question to both of you, actually. Maybe you could answer one after the other. Jyoti first. How do you look at artificial intelligence in your area of activity?
Yeah,
role of AI in your area of research.
Yeah, artificial intelligence can be used in our area of research and also Dr. Subi's, because we can predict the things like which type of molecules we should study, which we should focus in a particular type of nucleic acid target or a particular structure. So in that way, I think it can give us insights into our future work.
Okay, Subi, you want to answer now for this question?
Yeah, I think that is probably, I mentioned in the last slide also, something which is really we require is a kind of predictive synthetic sequence, predictive synthetic design if you want to go, especially in my field, I feel that if you want to go to address more and more complex structures, and we need to really look into kind of a predictive synthetic design. And probably that is one area probably artificial intelligence can help us. But I think, yeah, we need to work closely with them to get benefited from it. Yeah.
Okay, actually, for you, I think, Jyoti, they're not very specific questions, but many common questions. So maybe I'll first go to specifics to Subi. Subi, you were talking of self-healing. Where do you find self-healing applications? While you compared that cells have a tendency for self-healing and you're mimicking that,
yeah,
where do you see immediate applications of your work in self-healing?
Yeah, you can find many places self-healing applications. For example, you can think about you have a wonderful, beautiful car. You have a car paint, and if someone scratches the paint, and you can imagine if you are able to self-heal, or I mean, like if that paint is able to self-heal, there's a wonderful applications, right? So, and also any material, if you think about any coating materials, or even people are working on regenerative skins and all based on supramolecular scaffolds. I think in all those areas, I think self-healing plays an important role. And if you look into some of the industries, already self-healing polymers are already in the market. But they work on a kind of slightly different principle. For example, they have kind of this catalytic beads inside these polymers. Okay, and if you have a crack, and if they are able to find these beads, they are in situ. They can initiate the polymerization and self-heal. But one of the challenges with that is, if your crack is not finding that bead, right, you will not get self-healing. But the kind of polymers I talked about it's because of the dynamic exchange of the monomers, and so that kind of constraints are not there in case. And also you see that one of the video I had shown, there's a self-healing polymers. It's from a company called Suprapolix, and it's already in the market, the such polymers. So it's all based on these weak interactions and dynamic bonds, and they can self-heal. So it has interesting applications on various aspects.
Thanks. I think there's a question to both actually, because I think one is working on DNA, RNA kind of work, and one on smart materials, if I want to call. Why India is not self-sufficient on the fluorescent probes used for RT-PCR, and whether as Bhatnagar laureates, would you think of giving some quick solution on avoiding imports of these probes?
Actually, this is a very good question. That is, no molecular probes. We should make our own, not to import all the molecular probes that we use in the lab from the outside. So I think this is an area we should work together. Many of the young colleagues who are working on fluorescent probes, designing fluorescent probes, we should develop our own probes, or even we should commercialize the probes which are known in large quantities. So that's a very good question. We should work on this.
So do we expect Subi and Jyoti collaborate and give us a probe in the next 3 months?
Sure, several of probes like we have
For
designed.
honor we have given you.
Yeah. We can.
So I think this field is more appropriate for Jyoti, but any help?
Yeah. Yeah, like we have already developed a few probes that we can track the DNA and RNA in the cells, but we have to make it commercialized. We have to make it really, it should work in a cellular system. And selectively it should work. So these are the things I think we should work on.
Yeah, thanks. I think, Subi, you have another question actually. Can you throw some light on energy storage part? Will it compete with lithium batteries?
Yeah.
What would be the advantage is one question. Let me, because as I scroll again, the question will disappear for me. So next question to you again, Subi, was, I'm sorry, I have a question regarding functions of these polymers. Please specify what about DNA assemblies using polymeric vertices. So these are the two questions.
Okay, so good questions, and obviously, as you can see that what I was showing so far is we are trying to get a kind of more mechanistic understanding from a kind of design point of view. Although we work on functions, our functions are more towards optical and electronic, optonic and electronic applications. So coming back to your question on DNA nanostructures, we don't work on it, but then there are groups who work on DNA-based supramolecular polymers, especially in terms of especially the transient assemblies. Also, people work as a kind of carrier and delivery and so on. Okay, there are big groups working on it. Unfortunately, I'm not working on that. And coming to that electro kind of as a kind of energy storage, Again, a couple of my colleagues abroad are really looking into these aspects because we can think about this. One of the beauty of supramolecular polymers is that you can increase the order in your material. Okay, so unless you just kind of spin coating or anything of materials, if you are making a kind of electrode devices using some of these lithium materials and so on. But the beauty is that molecular self-assembly improves the order. And at least we see that in optical and electronic devices, this increased order has a kind of tremendous influence on its efficiency. I think in that respect, I think probably if we can think about the supramolecular polymers for a kind of lithium in a kind of batteries as energy storage, it probably that will help. That's my point. Yeah.
Thanks. I think there are not specific questions, but one question to both maybe. Maybe first Subi and then maybe Jyoti. Any message to young students who are watching us and researchers younger than you guys? I mean, so.
I'm sure the younger ones who are watching us are really smart. I'm like, they don't need my advice, but I would say based on my experience, I would say probably it's very important that you take up, if you would like to have a researcher, to continue as a researcher, probably you make up your mind at the early stages of your career itself and complete your kind of heart and soul into it, because that's the only way, I'm like, your dedication, if you are in a researcher field, it has to be probably easy that, I mean, it's important that you take up that decision at early stages. For example, when I took up my research career, I didn't know what is the career in science, because no one was really there to advise me at that point of time. But only my passion for chemistry drove me, because I qualified twice engineering examination. So it was a tough time for me to convince my parents I don't want to go to engineering and don't go to chemistry. But so my only advice is that take up your kind of decision to continue. If you want to be a researcher, take this decision at early stages of your career itself, and grab all the opportunities comes to your way. Don't hesitate. Don't be skeptical. So grab your opportunities. That's a simple advice. I don't know how it's going to help you, but I'm saying that's what I want to say.
Thank you, Jyoti.
Sure, I would like to say now you should have a passion for science and then for research. Any particular field you like, you can choose. Like you should love that particular subject and field. And then if you really continue and like the subject, then it's your hard work and perseverance that would drive you to do well in your future.
Okay, I think a question from me, I think maybe the last question. Geeta, if you have any questions, or I can take your question now.
Please go ahead. Please go ahead with your
tough question comes at the end.
Easy question. What next? I think we have heard of enough of smart material, self-healing, and click chemistry of triazoles of Jyoti. What is that next best cooking in your labs now?
So what we are doing, we are trying to do the dynamic combinatorial chemistry and more of this target guided synthesis to target, for the viral genome. We have already started, TAR RNA I was showing. So we are really trying something to do with the SARS-CoV-2 and also something else on molecular probes. And someone pointed out that we should have our own molecular probes rather than buying from outside from abroad.
Subi?
Yeah, so I briefly touched upon on this towards the last slide. I think what we should do, I'm like, this is our dream. I don't know whether we'll be able to achieve that, but what we really like to do is to go more complex systems, try to get fundamental understanding on this. We are still, but at very early stages of this particular field, we are just trying to learn what can be done with these structures. But obviously, we should go to more complex systems, and then our dream is to make real materials out of it. Obviously, there should be a purpose for doing all those things. Once we learn enough of it, I think we should put effort to extract function of these interesting materials, and probably of like a kind of one of the aspects I told about is kind of recyclable plastics. I think it has a potential opportunity for this particular kind of materials, and considering the environment pollution and so on, that's something which we seriously think about it at this point. I'm sure it will take some time before we really start working on it. And I think working on such societal problems is probably I should start. So far, I have not done it. Probably that's probably the future in my life.
Thanks. I think you touched upon circular economy, and I touched upon Atmanirbharta.
Yeah.
I hope your accomplishments in academic research activities which you have done so far hopefully would give us some light into circular economy, which you touched upon, and hopefully your depolymerization would become an important area of research for our country, and hopefully that would become the best green technology one could think of. And of course, the circular economy of taking every byproduct or side product into again a useful product. Hopefully those will be built into your future areas. And again, thank both of you for joining our invitation to be part of this discussions. And Geeta, over to you.
Yeah, thank you, Dr. Chandra, and thank you both for such excellent talks. I think it was a bit beyond my comprehension, but what I was happy to see is we started with chemistry and biology naturally flew into it. Like, I think we don't know where one starts and where other ends. We learn so much from nature. And I think these artificial boundaries which we create are only for our ease of understanding and the tools. But I think like science imitates art and art imitates science. I think similarly, you both have shown, extremely good fundamental science. And as Dr. Chandra said, I'm sure there will be applications of this, maybe not today, but tomorrow. And I think that's the beauty of this fundamental science. And we are able to see a lot of benefits, like Atmanirbharta will come when we have brilliant scientists working for our problems. And I think you people exemplify the best of India. And I'm sure we'll be able to work towards Atmanirbhar Bharat in every possible way. And I think the future is bright, and we are very happy to have three leading chemists of India today with us today to talk to us about the science. And I hope the audience enjoyed it as much as we enjoyed. And thank you all for being with us today. And we hope to come back again with the next round, maybe next week or later. Thank you.
Thank you. Thank you very
Bye-bye.
much. Thank you. Thank you.